You can see the slides, yeah.
Yeah, everything working.
Perfect.
Hello, everyone. Sorry for being late. but barely but. Hi, Matthias. Great to see you.
Yeah, hi.
I'm seeing everything works fine. Let's give ourselves a couple of moments before we start. Before we start, Matthias, are you fine with us recording this talk?
Yes, please. Yeah, if you can share the recording, that would be great. Then I also have the video. Thanks.
Yeah, sure. We are sharing it. We are sharing all the recordings in our directory, and in every reminder email, you have a login and password to the OnCloud directory where we have all the videos. Okay, and I guess okay, Irina is with us. Great. Let's get started then, everyone. It's 3 minutes past the hour. Whoever joins late, well, shame on them. And for everyone, we are happy to have with us Matthias Koenig, who is going to talk about modeling, digital twins, and pharmacokinetics. Matthias, the floor is yours. First, I will start the recording, but yes, it's already started. Beautiful. Okay, the floor is yours.
Yeah, thanks a lot. A bit of disclaimer, this talk will be very experimental and I'm trying to be very practical. I thought this is a bit of technical audience, so I can go a bit more in the technical details. And please feel free to interrupt any time and ask questions. It's much easier if you just ask and then I can explain certain things. Yeah, so the key focus of our research group is building reproducible computational models for personalized drug pharmacokinetics. And the key idea is to build computational models which can describe the vast variety of drugs which are currently on the market. So we started with a handful of drugs, but step by step we're trying to get more and more of the commonly prescribed drugs in our computational models. So on the one hand, we have this vast area of medications which are available. And on the other hand, we have this large challenge of intra-individual variability. So we're all different and everybody has different drug pharmacokinetics and different reactions to drugs. And now understanding how this mix of multiple drugs and individual variability come together, we're using computational modeling. So I want to motivate this with a simple example. So in this case, we take as a person a female, pretty young, and we take as a drug coffee, something you consume probably daily. And then we can look what will happen if we give a certain amount of this drug to this person. So in this case, it's like typical cup of coffee has around 100 milligrams of caffeine. And what we can then observe in this person is that we get a rise of caffeine in the plasma in around for like 1 hour, and then decrease over time. On the other hand, we can see that paraxanthine, which is the main metabolite of caffeine, appears in the plasma, and after around 8 hours also goes slowly down. Importantly, if you look at the individual data here, you can see a very large variability, and certain people have like very early, very high peaks in caffeine after drinking a cup of coffees, and other people have like much less concentrations in the plasma, and also the peaks appear much later. We are using so-called physiological-based pharmacokinetic models to now trying to describe these curves. What you can see here is like the experimental data. On the other hand, you can see also like some model predictions. And in the following, I want to present a bit like how these models look like, which can create these predictions of this clinical data. So what we're using is so-called physiological-based pharmacokinetic models. So the key idea is that it somehow rebuilds human physiology in silico. So somehow it's a digital twin of the body, consisting of compartments which are mainly connected via blood flow. These are normally ordinary differential equation models, which can be solved numerically, like given certain initial conditions. The processes which are in these models are mainly the ADMET processes, absorption, distribution, metabolism, and elimination. And we're putting this into different sub-models, for instance, often metabolism in the liver, excretion, elimination in the kidney, and transport and distribution via the blood flow, basically in these arrows which transport between the different compartments. These are multi-scale models and are also handled as multi-scale models computationally. So we have like the whole body circulation, and then we have sub-models for the different tissues which we can put into basically the circulation. In certain cases, we have even like multiple steps, like we have different cells which we put into an organ, which we then put into the cell, into the whole body. These models, personally, I like these models a lot because they have like high pharmacological and clinical relevance. What this means is basically on the you could use very simple models like which are phenomenological, and you have like 2 or 3 compartments, and you could also like describe such plasma concentration curves. But the main problem is that the parameters and the volumes don't really mean anything. They're more like effective compartments, effective parameters, and so on. And it's very difficult to describe pathophysiology or changes which you observe in patients in these parameters. On the other hand, here in these models, you have like a detailed description of the different organs and the different processes. And it's very easy to change certain parameters which correspond to certain real-world clinical measurements. Simple examples: if you want to build like a model for a small woman, you would change the parameters for the body weight and the sex, and use like sex-specific parameters, for instance, for hematocrit and so on, or organ volumes and so on. So there's often like data available which you can directly map for diseases or for different subgroups. Simple example how such a model would look like for this caffeine to paraxine example is that we have this whole body model. You could apply the caffeine mainly orally as like a solution, as a drink. This would then be absorbed by the gastrointestinal tract, mainly first go to the liver and then in the venous blood, and then be distributed via the circulation and go around. In the gastrointestinal submodel, you normally have like some processes for the absorption. Often this is a simple, simple first-order kinetics which transports the substances in the portal blood. And but there could be also more complicated equations. Then you have a submodel for the excretion. This is normally also like depending on the model, but it's normally also very, very simple. The drug is basically eliminated, and often this is also like mass action and first order kinetics. And then last but not least, you need somewhere the metabolism. Often this appears in the liver. Here the substrate caffeine would just via an enzyme be transformed to a product like paraxanthine, and this can be transported or imported, exported by the liver, and by that going back to the plasma concentrations. So like for to describe these curves, this would be like a model structure which would then be able to describe like this appearance and disappearance curve. So that's, I would say, the key idea of this whole things, and then it's very easy to study changes in parameters or pathophysiological conditions with this kind of models. I just want to give one single example for that. Here for the pravastatin, which is a cholesterol-lowering drug, where we looked at the effect of decreased kidney function. So what you normally do, like you have like parameters in the model which correspond to the condition you want to simulate. In this case, you have like a parameter which corresponds to the glomerular filtration rate, or how fast the kidney is able to filter basically the blood. And this is a single parameter, and you can just change this between 1, everything is like normal, to 0, basically the kidney can't filter anything. And then you can map this basically on the respective glomerular filtration rate, which you measure in certain subjects. You can then in the computer model very easily just change this parameter. Basically, you would change it in a range between 0 and 1.2, which corresponds to 0 to 120 milliliter per minute and body weight creatinine clearance. And then you can do a model simulation of the renal clearance of the substance. So that's basically the solid line is the model prediction, and then you can compare that against clinical data. So in this case, the model was built like basically for healthy subjects, and then this here is a model validation simulation where we predicted what would happen in renal impairment for this drug, and we had a like very nice agreement with the validation data here, which are these gray points and the points here with these arrow bars. Of course, with this computer models, you can then also like test very additional things. Like for instance, here often if you have renal impairment, you also have hepatic impairment. So you have a combination of hepatorenal impairment, and so we checked what happens if you have cirrhosis and in addition also renal impairment, and we can see like there's basically a bit of additive effect. And so if you have a bad kidney and a bad liver, you will get like very high area under the curves of the plasma concentration curve. And that's pretty bad because you want basically to be in the normal range. And so what you would have to do in such cases is basically a dose adjustment so that you if you're like very ill, you should take less of the drug because your liver and kidneys are not good enough to clear the drug from the system. That's the basic idea. And then you can do this for a lot of different cases. We did a ton of these models for different substances. So we have around 50 such models, like I don't know, 10, 15 publications about that. I won't go into detail, but we looked at a lot of different things which can affect variability, like pharmacogenomics, what is the effect of body weights, and so on and so on, with different drug combinations. But the key idea is always the same. You build this model and use some experimental data to parameterize it, and then you do some parameter changes, make predictions, and validate this with independent data. Okay. I keep this very short. If you want to know more, you can look at the references. There's also a lot of additional slides in the end, so if you want to look at it. But I want to focus a bit on the technical challenges and how to build such models and what are the problems, because I think this audience is like probably more interested in that than with me showing like 20 such curves and saying, okay, this is a nice model and so on and so on. Okay, so big challenge for this models is basically reproducibility, and like it's basically like everybody who tried to reproduce computer simulations is basically it's often like that here. Like you look at it, and on the first look it looks like pretty good, and the more you look at it, the more different it looks. So that's my personal experience, like in trying to reproduce code and simulations and models and predictions and whatever. So it's often like you get pretty close, but it's very difficult to get something which is the same. And the problem is like that there are a ton of bugs in our libraries and softwares and tools. And there were like some studies which looked, and normally you have like a 1 in 50 lines are buggy. And I don't know if this with AI and Copilot and so on gets better, but there's a ton of problems in our code bases. And most of these are very silent bugs, which normally don't appear, but some like if it's obvious, we know it and then we try to fix it. But there's a ton of things which is very difficult to handle. And so I want to show like some strategies which we use like to try to get down in this number of bugs and basically to be more reproducible with the data, but also with the computational models. So first part we have to handle is basically the whole data thing. And so in the beginning we were like, okay, let's just go to a nice database and get pharmacokinetic data, or look in the literature and get like a nice data set and so on. But the problem is there is basically nothing out there which was publicly available, or at least in 2016 and so on. And so we went and basically built our own database and started to collect data and digitize things and so on. And the main idea is basically summarized here in this figure. So we go to publications, we do put this in a standard curation pipeline. We then upload it to a database, having this process a lot of validation on this data curation, and then can use this via some API and programmatic access to build our computational models. We did this for a lot of substances. Basically, for the computational models we have, we established the data sets and can now work with this data. So how does this normally look like? You go in the publication, you look at the figures, you have to digitize basically these data sets here. You have to digitize the tables and put all the tables in, and you have to get all the metadata out of the study. So basically, what was the dosing regime? So how much caffeine did they get? And here, were they males? Were they females? Did they take oral contraceptives? Did they not? Did they smoke? Whatever. Basically, you have to go in and put this into categories and labels and get all this metadata associated with the different groups. And so in the end, you have like some curation of the parameters, of the time courses, and all of the dosing interventions and of the subjects. You can then do this for a lot of studies. Like here's a subset, like for one drug, chlorzoxazone. You normally have, like, depending on the drug, like you have a different number of studies, but around 10 to 100. And then you basically create that. You can see like all these different studies to different application protocols. Often they measure different things. They measure that in different subgroups, healthy, alcoholics, and so on. And then you can get all the data out and build one computational models which can use all the standardized data to try to make certain model predictions. Okay, this is just here to show, okay, from every of these papers, there comes one or two curves, but we have in the end one model which can integrate all the data from all the different data sets. Okay. We did this for a lot. Overall, we created like around 1,100 studies right now. I don't know, like 200,000 measurement points, around 10,000 time courses that we have, and so on and so on. And for certain substances, we have very good coverage. Like here's one example for caffeine, where you basically did a systematic literature research and review and basically created everything which is available and which we could find. And then we can use this data to basically do certain meta-analysis. Like this is one example for that. For instance, what you can see here is caffeine clearance against caffeine dose. Every data point is basically one clinical study. And what you can see is that you get a very large data cloud. But because we have all the metadata, we can use that to stratify the information. In this case, we can basically highlight the smokers in blue and the females who take oral contraceptives in orange. And then we can see like certain subgroups have a clear change in caffeine clearance. And then we can go back a bit to the biochemistry, and we see like, smoking induces the CYP1A2, which is a key enzyme in caffeine metabolism. So it makes sense that the smokers have an increased caffeine clearance. On the other hand, oral contraceptives have an inhibitory effect on the CYP1A2, which makes also sense. So, okay, women are somewhere in between. The women have low caffeine clearance. And then we can put this in computer models and then simulate, okay, what is with smoking women who take oral contraceptives, and then so on and so on. And so how does this look like? We manage this basically. So everything is managed via GitHub. We have like a repository, basically, where like a lot of people contributed over the last time, or the last 10 years. And then we have like certain tests on that, and then we have a large subfolder where we basically have the studies. So how do the studies look like? So basically for every substance, And every study we have like a subfolder where we have certain informations. So first we basically extract the figures which we want to curate. So here in this case, diazepatide, it's a GLP-1 agonist. We have certain time courses, and so we first extract the figures which we want to curate, the information about the groups and so on. And then we have two main documents. The one is a standardized JSON where we store the main information. And the other thing is basically that we put all the data into a Excel spreadsheet where we have the information for the different groups and so on in the different fields. And this is pretty standardized. And then we have validation in the database in the back end, which checks, okay, this makes all sense. Ages are not, I don't know, below 0 or like not above, I don't know, 150 and so on and so on. We have like interventions. So basically what was the protocols. And then we curate basically all the time points here from the data sets. Okay, so that's the main idea. And then you can use that and upload it to the database to see if I have this. And then we just have a script which upload that, and we can I don't know if I can show this quickly. Okay, so I have to open this here. And then we can basically upload this study to the database. So I showed like this thing. And then basically all this Excel and study JSONs and files are like uploaded and checked. Everything is tested that it's correct. And because we're using like clear, let's say like clear IDs for all for encoding everything, we can map this all immediately to metadata in the back end. So what I mean with that, basically we have like everything which is written here. It's basically defined and annotated to certain things. And as soon as we upload this to the database, this is checked and validated against like the respective fields. And then yeah, basically we can see, okay, the stuff uploaded and we have the time courses in there, and it has to index quickly, but the time courses will be available in a second to wait for the indexing. And what I meant like with this field, so basically if we, basically everything is annotated, and then we have metadata and ontologies and description, what does it mean, and so on and so on. And that helped us a lot like to get data consistency, and what ensures that the data quality is high is that we basically have like on the one hand this automatic scripts, always two curators go over this, and we have a lot of meta-analysis that we bring data together from multiple studies and find automatic outliers on things. Okay. So this, I would say, it's a short summary how we get the data for all these models. And the nice thing is that we have like highly controlled and highly standardized data for all the different models. And so we can easily bring different things together because we know, okay, this has the same structure and the same quality, and we don't have to worry about that too much. Okay, next part is about models. What we do in this part is like to ensure reproducibility is mainly using standards. What we mainly use is like systems biology markup language for the computational models. We use combined archives for the packaging and distribution, using like, I would say, not directly ZML, but a variant of ZML to describe all our simulation experiments. We use like this omics metadata to annotate all our information. And for the parameter optimization, we use PETAB. SBML is very nice because it allows us to solve a lot of different things. On the one hand, it has like very nice encoding of the ordinary differential equation system and of this process-based computational models. And that's basically what we have here for this physiological-based pharmacokinetic models. On the other hand, with SBML comp, we can easily build hierarchical models and have basically submodels where we only describe the interfaces and the mappings and deletions and replacements to couple the model. So like basically we can have a lot of different submodels, and then we just say what is on our interface and bring everything together. It has the annotations, and so we can ensure that our models have the same annotations like the data in our database. And so like we have a lot of scripts which can do automatic mapping of data to models. And we have especially unit validation, unit checking, and so on, which ensures that our models have like high quality. Key challenges, I would say with ODE modeling for us is like on the one hand, like the programmatic model generation, which is often like a bit, it's a bit difficult. Model visualization, model communication, and dissemination of our computational models. And so in the following, I want to show like the solutions which we use like for these different problems. For the packaging, basically we use PyMetadata, which is a tool which we wrote which supports basically combined archive and allows to handle all the metadata, basically the RDF triples, and resolving the things using like identifiers.org and information from the ontology lookup service. And so then we have a simple Python library which allows us like to create this combined archive and then add entries. So basically add different resources in here, mainly computational models, but also figures, descriptions, simulation results, and so on. And then we can just package this in this omics, which is, I would say, a fancy zip archive with a bit of metadata. For the model creation, we use basically SBML utils, which is I'll say like a Python abstraction layer around libSBML. So libSBML has Python binding, but it's a very low-level library to interact with SBML. We wrote basically a more like high-level wrapper, which makes it easier to build the computational model. That's also often the problem. You have like the standards, and then you have a very low-level library, and makes it very hard to do the actual tasks which you want in modeling or visualization and so on. And so often you have to write like a more like an abstraction layer on top of that to do your actual modeling. And this is what SBML utils is. It's provide like this higher level thing, which has a lot of like this everyday stuff that we need. Like for instance, one thing that we really wanted is like a markdown annotations on our informations, because we use a lot of markdown to document things, but we want to have directly the markdown serialized in the annotations and in the metadata of the SBML. And so like for instance, this was also solved here in the library. Also things for model creation, model merging, model manipulation. Third part is I will quickly run through the 3 or 4 tools, and then I will show a quick demo how this normally looks like when we build the models. So SBML for humans is basically a tool which allows you to, I would say, like to disseminate your SBML models. We often have the problems that we have collaboration partners. It's very difficult to explain them the models and to communicate with them, because they can't read XML and they don't understand equations, and mainly clinicians, not dissing anyone here, but like they have a different expertise, but they're not like computational modelers. And so often we need like a more high level way for them to explore what is in the model and to understand it. And this is basically what's happening here. It's a web tool where you can just upload your SBML, and then it's rendered nicely, and you can interact with that, and especially you can also filter it and search in it, which is very helpful for us, because often we work with these larger models and we don't even understand ourselves what it is in there, and then it's like, oh, this parameter, what unit does it have, and what does it mean? And then you just look it up in this interactive report, and then you're good to go. Next thing that we use a lot is like SciSBML. This is basically a cytoscape plugin for SBML. This allows you basically to get more like a graph, I'll say like a classical graph or network, more like network view on your SBML model. And especially as it has different layers, often what people only see is the species reaction graph, but we also like have certain things like that. We have a marked graph. So basically you have your, okay, this is, if you don't understand the words, ignore them. If you understand them, you probably know what I mean. But in your computational models, if you have math, you have an AST node tree and AST node dependencies, and you want to see them. So basically, you want to know which parameters affects which other AST nodes, so as an information flow. Because it's often important, you change parameter names and so on. And if you then have isolated nodes which are not connected, it means like your parameters don't appear in the right math or at the right positions. And so this allows you to navigate basically through the AST node tree and these connections. very helpful if you're like deep into modeling. Otherwise, just ignore what I said. Okay. Yeah. And so how does this look? How does this look when we build the models? So basically, we have also our models. They manage in a repository. So that's basically a different repository here. And there we have the different models which belong then to the data you saw before. And we have a subset where we build the models. And as you remember, we have this hierarchical model structure. And also like here, we have this hierarchical model structure. You have a model for the body, intestine, kidney, liver, and so on. And then we use SBML utils here to basically define a model object. We can add compartments to that. We can add species in the compartments, and we can add the reactions. We can also do layouts and annotations. And currently, like, okay, I wanted to have a demo for that, but we're also working on automatic SPGN conversion. Basically, you can use the SPO, again, very technical. If you don't know what I say, ignore it. You can basically use SPO terms and annotations on the objects to automatically get an SBML to SPGN transformation and create maps out of SBML models. And so basically, like we're currently implementing that. Other tools also did this. And I think Minerva or like Mimosa and stuff like that did something like that. But that's a key idea, that you can have layout information in here and with the annotations automatically create graphs. Okay, so how does this look like? We can then execute that, and basically what this is doing, it's creating the model, and we can also directly connect this to the visualization, so that we can explore this with this SciSBML. So if we, okay, sorry, I have to wait until this starts up. So if we run the model, basically it's going through this here. It's doing a lot of validation if the mass and everything is correct, and then it creates like basically the visualization with annotations and so on here. So basically this is like just the liver submodel where this tirzepatide metabolites are imported and they excreted in the bile. So that's a very simple, okay, this is just transported through the different compartments. But on the other hand, you also have like this more complex kinetics graph where you now can see, okay, which parameters depend on what is connected to which plasma compartments and so on. Okay, so that's the core idea. To bring the models together, you basically have like then mappings and interfaces and ports. And then in the end, you can basically build all the different models and create all your files in one script, and then use the combined archive and so on to basically store everything. So what this did now, it's basically created all the different submodels, the kidney, the liver, intestine, the whole body, and then it flattened it to one executable model, put it into a combined archive, and we can see basically here what was packaged in there with the metadata, and in the background it basically did the visualization of the whole things. And then you can see here now this all looks more complex. So normally these body models look often like the spider webs, because you have certain substances which are circulating in these models, and then you connect them via the tissue models. Okay. And then, probably you don't see much here, but you also have access to the metadata and so on, and can map it and explore it, and could map, also like map simulation results on this whole visualization. So that's the main idea. You have this model, and then you can then also automatically, you could upload it then to SBML for humans. How we distribute normally our models is that we build a separate repository for that. We then link the repository to Zenodo, which is a very nice trick for people. If you want DOIs for, if you want to versioning things, you can do an automatic Zenodo GitHub integration. Then you get like DOIs for your releases. And then we can basically provide the files in here, and then link automatically to the report. So we could go to SBML for humans, like basically the site here, and then upload this, but you can also like just somewhere put the URL and say, hey, please upload this model and integrate it here. Okay, so this is the SBML for humans. So this is this high-level report for the same model. And I would say the nicest thing, or how we use it mainly, is basically using this filtering option here. so that we can just type something here and we only see the objects in the model which are related to this thing. And that, because if the models get very large, it's very difficult. And so that's, I would say, the most helpful thing. And we can also navigate between different things and see the math and especially see the units, which we need very often to make sure that our interfaces and ports are correct and everything works. Okay, so that's the part. And normally we distribute that, and the more everywhere bugs, like I said, you can see here error 404 and whatever. But yeah, the more metadata we put here, the easier it is for our collaboration partners to understand what's going on with these models. Okay, this is the part about the I would say mainly the SPML workflows which we use and the SPML tools. On the other hand, we're working also on a bit on SPGN, and I don't know, many years ago we basically did a Python library for SPGN. Some tools use that, and it's very useful. So if you're interested in working with SPGN in Python, have a look at the libSPGN-py. Actually, like until this disease maps meeting, there will be a Maya update. We will have like a very nice up-to-date modern like data classes and so on, and you get like very nice code completion and like type checking and whatever you want. But there will be a very new version around the corner, and we will also like have like certain features like SPML to SPGN conversion. So if you work in Python and with SPGN, this could be something nice for you. That's that. Yeah, last but not least, yeah, but we're working on mainly only also like with Irina, with Dagmar Waltemat, it's like making our models more fair. And this is a bit complicated. Often like people just call their models fair. We try to really do like a fair evaluation of the models so that we have certain indicators and we can check, okay, man, do our models fulfill these indicators? And then we can use this evaluation of the models and the indicators to basically get a feeling, okay, where could we improve our the fairness of our models? Like here, I want to show an example of one of my models, the dapagliflozin, and then you can have certain indicators here. Like, is a model identified by a globally unique identifier and so on? Can it be accessed manually and so on and so on? And so, like, if you provide your things via nice repositories and with licenses and so on, you will get more points on here. This is not like some, I said, like some absolute scale. It's more like, I would say, it gives you hints where you could improve the fairness of your models and your resources. And so we basically did this for our for the dapagliflozin model. It's not too complicated to do that. It takes a bit of time, but you basically fill out an Excel sheet, which gives you an overview of the of this information. To quickly check, find the file. This work? Sorry for that. Okay, here. And there you basically have certain indicators for findability, accessibility, interoperability, and reusability, and then definitions and explanations. And there are already existing some examples from biomodels from the Physio repository, and like also here we evaluated our GitHub based models and gave these indicators. Okay. Yeah. Last thing I wanted to show is before I go a bit in the future is basically this SPML sim. This is basically in the end what we're using to run all our simulations. Can basically say don't use that. That's what we use it all the time, but it's like, man, this is if it's like basically alpha and no documentation and nobody knows what it is, and not even we, but that's it's basically our workhorse to do all the simulations and do all the information. And I just want to show in the end okay, how this normally looks like. So we have these models and we have all this data, and so what we do, we're defining these experiments. which basically tells you what do you want to do with the models. And this is, like I said, like some variant of ZML. So you define basically, okay, which data you want to load. You define certain simulations, and basically you tell it, okay, what kind of time courses you want to run and what model changes do you want to do. Like for instance, if we simulate a certain study, we have to set the body weights of the respective cohorts. We have to adapt the dosing scheme for the whole things. And then we can basically map the data on the actual model output. So you have observables in the models and you have data you have to map on that you can do automatic parameter optimization and so on. And then in the end, we define basically some figures to visualize that. And then normally we can run this through, and then you get basically the plot. And okay, this was a model simulation with the respective data. Yeah. And then we do this basically for all the data that we created for a certain substance, and then can run all the simulations and all the parameter scans and so on. And then we will get basically, okay, this model performs on this data sets like that. So it now runs in the background all the simulations. And then we get also like some very nice reports, which, like I said, it's not really ZML, but it's very, if you know ZML, you know what to do with these results and the reports. Sorry, this takes a bit. We have to do, we basically do like a lot of parameter scans now in the background, which scan like renal impairment, hepatic impairment, and so on. And in the end, or here the body weight dependency of the drugs, calculate pharmacokinetic parameters, and so on and so on. But the whole idea is in the end that you just press one button and it gets all the data from the database. It takes your models, simulations, and you get a nice report where you basically have all your, this were all my simulations and all my results. And if I'm interested, okay, what did actually happen for this? You can basically go in more details and see, okay, this was the code which generated it, and then you can immediately see which parameters were changed and so on. And then we can distribute this here with the actual models. I have to show. So for instance, you can then just put the simulations in the repository. And so normally we do now one model version, and we put in like the results of our parameter optimization, and we put in our automatic results of the simulations. And then you have basically one model version with which we can easily run all the simulations and also have the information about the parameter optimization in there. And then you're sure like everything belongs together. Because a big problem is always like that you get out of sync between plots and simulation and so on. So our thing is basically we create just all plots on the fly. Don't save anything. You should always be able to press a button and then everything works. Okay. So, but this is basically done via this very experimental stuff here. Okay. So in the future, where do I see to go there? Or where do we want to go? The one thing is definitely we go currently towards multimodal digital twins or computer models. So basically you can see like we have this, we did this for 20, 30, 40 models in isolation. And now it's hey, let's have one model to rule them all and put basically, let's put like the data from 1,000 papers and like from 40 models together and have one thing which can simulate all. And then have like at certain points, basically drug-drug interactions and simple basically you have transporters which transport certain substances, and so you have like some competitive inhibition and so on. So we would like add like a few like linking points between all the substances, and then you could say, okay, what is happening to a 80-year-old person who takes statins, beta blocker, diuretics, and now also GLP-1 agonists? And what are the interactions? Can I simulate this individually and so on? So this multimodal integration. So that's one thing. The other thing that we do a lot, I didn't have time to show that, it's but more like towards spatially resolved models. So what we do now a lot is like this whole body scale, and basically have like tissue, like cellular scale, I would say it. We scale it up to the tissue, to the organ, but in the end, it's like simple pathway models, which we put into the different organs. And now we go more and more towards more detailed description, that we have actual data about vascular networks and about the microcirculation and so on, and that we can put this into a multi-scale model to simulate, okay, what would happen in the different organs, like spatially resolved. because there you have a lot of heterogeneity, and to understand that is definitely a second thing that we're working on. That's basically it. Yeah, sorry, this was a bit probably a different talk than you expected. I wanted to show a bit of like basically just walk you through. And I know like certain points you can definitely take something away for different things, but it's more like to show like how we achieved like over the time like to ensure reproducibility and also like say easy of use. Yeah, because in the end, we have like a lot of students who work on this project, and we have to somehow onboard them and bring them to speed, and to ensure that everything is on the same page and works together. And this is all, like basically by embracing standardization and having our standardized workflows and going through that, we can ensure that. And also like students can very fast build these models and do the data curation and have high quality simulations. All the slides are available, so just scan the code if you're interested in that, and also if you have questions, yeah, you can ask me offline also, or now.
Thanks a lot, Matthias. That was a very, very interesting talk. I have plenty of questions, but as I'm not selfish, I'm okay. That's Robert who would like to ask you something. Robert, go ahead. Robert, you're muted. If you're asking us something, we cannot hear that. Okay, now it's better. Let's try.
Matthias, I wondered if you could say a little more about the metrics you use in the interoperability part of your evaluation.
Good question. I'm also not an expert. It's you have to. Irina is definitely the expert in that. But here, interoperable, like it's basically archive content is expressed in standardized formats. The model content is expressed in standardized format. So this is basically coming from RDA indicators, and they are mainly Irina and Dagmar, but also like a whole working group, try to adapt them to computational models. What does fairness mean for computational models? And so this is like, yeah, so metadata is expressed in standardized formats and so on. It's machine understandable. They use fair compliant vocabulary and so on and so on. So it's basically oriented at the RDA. I honestly have to say it's difficult to say what does it mean for my actual model? Like what is fair compliant vocabulary? So like, and I would say like, okay, man, RDF triples are good, and then there are standard ontologies which are used in your fields, and if you map to them, I would say you're good. But that's not like how I say, like, it's not like a checkbox. It's more like, okay, I would say like, if you want to know what it means, then you understand it, but there's not a clear, you have to use this ontology, you have to use that. And I also don't think this is what this should do. It should give you more an indicator, okay, I'm not using any ontologies, I'm not using any vocabulary. Probably if I should do that, then I'm getting a higher score on that, but it makes also my model more useful to other people. So I see this more like that.
Thank you.
Okay. Any other questions to Matthias? Okay, then I will profit. Okay, let's start from the beginning. PBPK database. Do you use any specific ontology or dictionary or vocabulary to harmonize clinical vocabulary describing different studies?
That's complicated. I would say how I see it. What we tried. We saw this is very complicated. I have to go really back to answer the question. We saw this a very complicated task, and our main goal was to build a database which is very extensible. So we didn't want to have like hard-coded tables of what information should be in there, but we decided we have like basically an extensible ontology in the background where we can say, okay, this we can use in certain fields. And so we have an ontology, and then we say like, okay, certain things can be used. And so this is basically our main ontology, which describes what is allowed in the database. And then we map the terms of these ontology to other ontologies. So the idea is like by that, and then we have our own thing to describe everything in the database, and then we can reach out and say, hey, this age term, it's the SNOMED and so on term. And by that, it's also like we don't have to complete with the mappings yet, but step by step, we can get more terms in. Personally,
Okay.
I find it very complicated because all this SNOMED and stuff, it's often like it's not easily accessible. Like I'm used to biontologies and ontology lookup service and so on, and you have like a nice ontology you can download and work with. But this medical ontologies, they are somehow proprietary, and you can add the terms, but it's very difficult to work with them. So for us, it's more like at some point we've solved this problem, but it's yeah.
I think you kind of pointed this out that the clinical researchers have a different role to play, and it's also from my experience that the medical ontologies are difficult to apply. And yeah, I just wanted to know if you apply anything, or is it, for instance, if I would be interested in you know, applying something, it's up to me to curate. It's fair in any case. If I'm interested in, I don't know, using OMOP, then it's up to me to take this, but you give me all the information that I need to do that.
Exactly. Like for most things, like simple chemical molecules, I would say they're pretty good annotated and they map out to everything. Also other things like organs and stuff like that, or tissues, yeah, we basically just map to the standard ontologies. I even forgot the name, FMA and stuff like that. And so at least if you know what you're doing in the ontology field, it's very easy for you to get a mapping. I would say that. But I don't know, it's complicated. For me, I thought like these ontologies are very useful, but I'm not. I don't know. I haven't seen much use cases yet. Like for us, the main thing is we annotate our models with the same terms like our data, and this allows us to do some automatic mapping. But that's because we have like some control over both things. Then integrating with other ontologies, it gets much more complicated because often it's like, okay, how do you map all these ontologies to each other, and what does it mean? Is it
Yeah.
you know all the problems, yeah.
Yes, so classifying molecules, that's one thing. I was also thinking about, you know, description of clinical studies that were used to feed the database. Because I remember clearly when our group leader was explaining us that he was in a TC where for an hour and a half clinicians were arguing how to measure weight. And basically, and it was diabetic weight loss. One would understand this as something natural, but it would be, okay, is it delta over the study? Is it the weight before the study? Is it the weight after the study? Is it measured like this, like that, in intervals?
It's a good point. And we see this also like in the reporting of the studies. It's very heterogeneous. Everybody reports something else. And so our strategy on the one part is we have this ontology to describe different things. On the other hand, what we saw is that we use a lot of like combination of fields. Like for instance, we have a measurement type which is concentration. We also have like something which is concentration change, percentile change, and so on and so on. So we have like a lot of these different terms where we can internally convert them and unify them, but we curate first everything which is in there, and then we have rules how to match things on each other. And then we use combination of, okay, like for instance here, the concentration was of like for instance, we don't have a measurement concentration HDL, but we just by the combination of, okay, this measurement was concentration of triglycerides in the plasma, and the method was not reported. Basically, this gives like by combining the things, we get basically we can cover much more. And so we try to put this into the smallest possible pieces, and then by combining that and using the ontology, we pretty flexible. But good part is pharmacokinetic studies are very homogeneous. We applied this also a lot of like as soon as we go to pharmacodynamics and so on, man, everybody's reporting such crazy stuff. You can't have one database to rule them all. But we're trying to what's easy to describe, we can definitely describe here.
Yeah. Okay. Yeah. So if you don't mind, I would have a couple of more questions. It's I was wondering about the model deposition, the idea about the Git integration with Zenodo is cool, but do you deposit this on biomodels at all, or?
I don't like. Personally, I hate the CCO licensing of biomodels. So basically, if I put it on biomodels, everybody can take the model without citing me. I really don't like that. That's not a good thing. Like, it's basically, if it would be CC BY, or I could get an exception for CC BY, yeah. I see also the other point, it's really problematic to have not CC0 on databases, because then it's really hard to work with the different data sets together. I see both sides, but like as me, it's like my main job is not developing databases. My main job is developing models, and I want somehow citations for that and acknowledgement. And so I would say that's the main reason. But often we also put it into biomodels. Most of our models, like, yeah, I'm not like I just personally I don't like the licensing, but okay, that's it. But yeah, we deposit also in biomodels. The point is often yeah. Probably it's not clear how to do everything right now. And what I think in the end, like we will have like a large combined archive which we create with all the information and simulations and results and parameter optimization, and we would deposit that to biomodels, and that biomodels get more and more support for additional assets. And they do that. Before it was only SBML models. Now they're much more flexible with the model formats. They want to go to AI models, and so I see that this more like, okay, this has to be a whole package, which makes it more fair and more reproducible. And so I would see that what we have in the Git repository, we would probably deposit it like one-to-one in the biomodels. And it would be hopefully also the idea that in the end we basically just have a GitHub action, which would say, hey, finally, biomodels, you have the API that I can automatically deposit, and then we would work with our GitHub workflows, and we would just have GitHub actions which deposit this everywhere and map it and whatever. Yeah, that's a dream.
Yeah, the CC0 point is actually related to my other question. It's you're building this fantastic ecosystem of multiple models. I'm just wondering, and this is a very honest and practical question. Have you ever reused a model of somebody else?
Of somebody else. That's a
Yeah,
good question.
that's
Yeah,
not from your team, not by somebody
but
you have
definitely
trained.
simple models. I would say yes, but it's simple models. A lot in the context of pharmacodynamics. A lot of models like for instance, oh man, the HbA1c turnover, erythrocyte turnover, and glycosylated hemoglobin. And often, like, we have a model for blood glucose regulation, and we couple just to the pharmacodynamics part of that. Other things, submodels of glomerular filtration, that they have more detailed kidneys, we put them in. Also pharmacodynamics weight loss model, for instance, that we had a weight loss model coupled and so on. But often this is, okay, the models are not available in standard formats, and they're very simple, 3-4 equations, and we put the equations in SBML. And it's not like that we downloaded a file and then it worked. We found the equations weren't too complicated, and we re-implemented it and then coupled it.
Okay. And my last question is how your normal workflow looks like with expert validation of your models. When you are finally getting a domain expert, you are finding I don't know, half an hour of their precious time, and they are sitting together with you. Do you show them SBML for humans? Do you show them the network in Cytoscape? How does this look like?
I would say often SBML for humans, I would say the main challenge is that we don't understand what the clinicians want to see, and they expect certain variables and certain readouts which they normally see in their clinical context. And often these are things which they get from their clinical data, clinical reports. And so what we learned is we have our normal model readouts, and we have to map them on what they know from the clinics. And so, and often it's like this layer of communication, and there we use a lot like SBML for humans and so on to say, hey, look, we have in our model this parameter, and for us, this corresponds to that. And what do you normally measure in the clinics? Or is there some assay to get information about that? And then we understand often the assay, and we add like a small things to bring that there. For instance, in the beginning, we had just a parameter which was called F kidney function, and it was between 0 and 1, and they were like, hey, what is that? We know glomerular filtration rate, and so we just did a mapping of the two things. Okay, and instead of a 1, it's 100 now. Okay, but but the clinician understands glomerular filtration rate. They have no idea what we meant with this parameter. So I would say that's finding this mapping of what does the model mean, the model variables, and they say what is useful in clinical practice and or in our guidelines, and then adding this mapping layer to give them some output they understand. Yeah.
Okay, awesome. That would be it for me. Are there any other questions? I know we are at the hour, but we can still go by. Robert?
I'm very happy to get questions.
Robert, please go ahead.
I think that maybe another sentence to be said about the mapping between what the clinician expects and what the model has predicted is that you don't want anything to get lost in that conversation. It's important to have somebody on the clinical side help you with the mapping. Somebody smart, of course, somebody who's thinking about the basic science. But I've found that one of the most important roles we play as modelers is to make sure that nothing is falling into the gaps as we work with clinical investigators.
No,
in other words, you want the mapping to be correct.
Exactly. That's but I have also say that the most complicated part is like getting this towards the clinics. And then one part is definitely the or biggest challenge is the heterogeneity also in the clinical data. Like a lot of these models are built like with retrospective data and like from publication and digitized and so on. But if we have to work with the actual clinical databases and the clinical data sets, it's often like, I mean, they're very heterogeneous and they're very sparse and like lots of empty things and so on and so on. And so it's often like, we have to talk early what can actually be measured and what can be an input of the model. And but I
think I'm just advocating for communication across the boundary.
Yeah, that's for sure. Like but it's I think we as modeler we really have to understand what the clinicians use in their decisions. And I think it's on the one hand, okay, this is important. On the other hand, I think also with this whole AI and automatical and clinical decision support and so on and so on, this is probably getting less and less important because I can see, okay, what you have right now is we still have to communicate our models to the clinicians. You know, we have to make this intermediate layer. But I see a lot what will happen is that we use clinical data as an input to our computational models. This model predicts something, and it doesn't really have to map to the clinical things. It's basically a new feature generator, and these features can go with the clinical data together in a clinical decision support system, and AI can figure out what it means. So you basically don't have to map it really to get better to get better clinical decision, and the clinician only sees the output of the AI with like, I don't know, some feature importance and so on. So it will be more the problem of like communicating AI and explainability and all this stuff. But I would see like this more like deterministic models and like models built on real world equations and insights. They will get more and more like just feature generators for AI systems. So that's but we don't probably have to do a bit of mapping, but it's like probably we don't have to care so much about that anymore. I don't know, perhaps not, who knows. Yeah.
Thanks.
Thanks a lot, Matthias. It was a great presentation. I think that the fact that you went that you took us through the tool set that you're normally using, that was awesome. That was a very great hands-on walkthrough, and I benefited a lot. So I hope others as well. So I'll thank you very, very much again. And See you next time, everyone. It will be in 1 month again, because in 2 weeks we are going to have, I think, likely an open mic. I won't be able to attend, but I guess you will be feel free to join. But in 1 month we are going to have another talk scheduled. Thank you very much again, and see you around.
Yeah. Thank you. Bye.
Thank you.